Hi everyone, we are in another lab tours in the Photomicrobe group, and here we have Elia, which is going to give us a nice interview. So, hi, can you talk a bit about yourself, about your research?
Of course. I'm, as he said, Elia. I started working here about 8 months ago in August, and I'm working with cyanobacteria mostly, but all kind of photosynthetic microbes are used in our group. We do basic research and applied research, and we study photosynthesis and how to apply it to make the world greener.
Wow, that's very cool. And what is your research topic specifically?
I specifically work on producing solar butanol. Butanol. It's a chemical that can be used as a substitute for gasoline, for example, or in the chemical industry to make packaging as a feedstock. And basically, I am trying to develop a new photobioreactor that involves immobilizing cells in a gel-like matrix and producing butanol for this.
Well, that's very cool. Like, it's very important, of course, to make the world greener and to have all this research towards a more environmentally friendly fuels and everything. That's very cool. So, do you, for example, engineer bacteria or algae? How does it work? Or are you focused more on the because if you're doing like more bioreactors, I think you're more on the, let's say, on the environment surrounding this algae than the algae itself, right? How is it?
Yes, correct. I focus mostly on engineering the photobioreactor, so the environment in which the bacteria grow and produce butanol. But in our lab, we are working also, my colleague are working into engineering the photosynthesis of these cyanobacteria in order to improve the production of butanol. And I'm using an engineered strain of Synecocystis produced in KTH in Stockholm by Professor Hudson, because Synecocystis does produce butanol normally. So it has been engineered and it's producing butanol right now.
Wow, that's super cool, and this collaboration is like so important because each group is going to have like one specific focus, which is very nice. By the way, did you have like are you more from an engineering background? How is it?
I studied biology in my bachelor, but then I moved to biotechnology for the bioeconomy in my master. It seemed a very cool idea, and I still have that idea. And so I'm in between the engineering and, let's say, biological figure. But right now I'm more on the engineering preference.
Cool. And how was the process of you joining your lab, your group? How was it? Where did you find about this position?
So I was working in Italy in a startup, but I missed the excitement of research. So I started looking around on LinkedIn, on Find a PhD and AgriStock. And I came up with the job offer from Professor Jagot. In this group, Finland seemed very interesting. AgriStock only has very interesting offers in the field of agri-food research. So I decided, why not? I applied, and after a screening process that took about 3 weeks, I've been offered a position to start a few months after. And that's it, yeah.
Wow. Yeah, that's interesting. I think you're the second person that mentions finding on LinkedIn about this position. So yeah, I think many job placements are advertised on LinkedIn also. Why do you think startups were not like your thing, or maybe they are your thing? But how would you compare a startup and actual academic research?
So it depends. What kind of startup are you talking about? Because there are some that may be focused on research, and some that are more already have a product and want just to have the scientific person to validate their product. So where I was, it was the second one. So they had the product, they wanted to sell, and they wanted me to help them. Mm-hmm. So what I see different, there is a good degree of selling your product in research in academia, because you need fundings, and with fundings you have to sell your idea. Yeah. So it's not so different, but you have the freedom of experimenting with a lot of equipment that in many companies you cannot find. because they are focused elsewhere.
Yeah, of course. In academia and research in general, we have much more freedom to experiment. Whereas in a startup, of course, you have to narrow down like a single product. Yeah. Wow, that's very cool. I'm really interested in like all these things you have here. Would you mind like showing some stuff that you have been working with?
Of course, I can show you a few things. For example, is a gas chromatographer, and I use it personally to measure butanol, and you can see the result of the measurement on the screen. There is the solvent, the internal standard, and the interesting molecule that I'm looking for, that is the butanol.
Oh, but it's so tiny.
Yeah, it's so tiny, but actually this is a huge peak because I put a lot of internal standard to be sure that it's always there with the same intensity. I can show you also, for example, if you follow me.
Yeah,
I
of
can
course.
show you our culture collection. We have about 100, maybe 200 different strains here. Some are natural, some are GMO. And all of them are right now not used, but maintained by our employees, colleagues. And about every month or a couple of months, they are refreshed in order to be readily available when we want to use them.
Ah, okay. So this is more of a like a storage.
Yes, this is a storage mostly.
And refresh so they don't die, of course.
Exactly.
So you're putting more nutrients in the
More
medium.
nutrients, more mostly water and nutrients in order to have them survive enough till the next use.
Cool. And there are some red ones there, right?
Yes.
Cool. Not all algae are green, I think.
Exactly. Not all algae are green, and not all bacteria are cyanobacteria. So in this case, it could be, let's see. This one is hematococcus, so red, hemo, like the blood. And yeah, this is hematococcus.
Wow. This is so cool. So they have like kind of a bacteria algae zoo here.
Here we have all the cells that we are using in our project for solar butanol. And as you can see, there is my colleague strains that he's creating to improve photosynthesis, and my strains that are trying to produce butanol. They are doing it, and they are grown in 1% CO2, so a lot higher than what you can find in the normal air. CO2 will push accumulation of biomass and products toward higher titers. What else can I say? These bottles here on the right are being bubbled with CO2 in order to be more productive, accumulate biomass faster, and to be mixed, because these bottles are amazing for light penetration into the culture, because being with a good surface to depth ratio, the cells all get almost the same amount of light, and not like in a flask where only the cells that find themselves right closer to the light have.
What is the efficiency of bacteria getting transforming into electrons or something?
Generally, efficiency of photosynthesis is very low. The proportion of light hitting a leaf or a bacteria that is actually absorbed and transformed is in the order of probably 1%. I'm not sure of the number, but it's in the range, 1-2% maybe. So it's not really efficient. But in our cases, we use very low light. Probably in this case, it's about 100 micromoles per square meters of light. And it's about 1,000 of what can eat the ground in a normal day, in a normal sunny day. So we use low intensity light to not strain and stress the cells.
Wow. Yeah, like in comparison, I think the cutting edge solar panels nowadays are around like 30% of efficiency. like something. But of course, it's very different because here we're dealing with living organisms, and there's a huge supply chain for you to manufacture, let's say, solar panels.
Yeah.
It generates a lot of waste. It generates a lot of environmental impact. Whereas like algae and more living organisms, I think it has a much simpler, let's say, manufacturing process, if you can say that.
It can be simpler. If you want to accumulate biomass, for example, you just need a big pond, some water, and some sunlight. And microalgae will start growing. It won't be efficient, but it's definitely cost effective.
Okay, yeah, that's very interesting. Of course, especially if you have a lot of land that you're not using, right? Yeah, but cool. Thanks for showing your algae zoo, and maybe we can go somewhere else.
Yes, for example, I can show you how I can immobilize cells for my experiments. In this case, I will immobilize some chlamydomonas in blotting paper. Blotting paper is a good support for immobilization as it provides a porous environment, non-toxic environment, and a stable matrix that we can put in water and it won't dissolve. So,
why do you want to immobilize them?
Immobilization is useful because being in a porous environment, the space is limited, so cells can divide only a few times and then stop and focus on producing what we really like. In this case, it could be hydrogen from chlamydomonas, or from Synechocystis that I'm using, it could be butanol. So immobilization will allow us to shift the metabolism from growth to products.
Cool. So cells have some inner states, and sometimes they're focused on more like one thing or another. And yeah, he's trying to make them focus more on energy production. That's so cool. That's so cool. Can we see a bit better?
Of course.
It's so interesting. So it acts like an ECM, right? Like more or less of an extracellular matrix.
Yes.
Yeah, that's so cool.
It should work something like similarly, but it's more controlled than letting the cells doing film on their own, and it should be more efficient. So in this case, I will be putting my cells mixed with alginate. Metal bar will allow us to have a specific thickness of 200 microns, and I have also to stop and to cross-link the alginate with a solution of calcium chloride that will put itself in between acidic residues in the alginate and create a bond, kind of a bond that will allow the matrix to stay still, like a gel, like a gelatin, and prevent also, again, the cells from growing even more.
Wow. Thank you so much. It's so interesting.
Producing like this, then we can put it in a continuous flow bioreactor and grow it under specific light condition, specific temperature condition. We can play with the pH, we can play with nutrients availability to increase even more productivity of our products.
Can we see it?
Of course. Follow me, please. Here we have a very simple and very homemade photobioreactor. This has been built by our researchers together with our machine shop of the University of Turku. It consists of glass panels held together by silicon, and inside there is a foam support that will allow water and water medium to flow upward via capillarity through to the blotting paper containing the cells. The cells here will have all the nutrients, all the CO2 necessary to grow and produce. In this case, they may be producing hydrogen because we are using chlamydomonas. But in case of other microalgae or cyanobacteria, they may produce ethylene or butanol if they are engineered so.
Wow. What is this sample producing again?
This one, it's a Chlamydomonas strain that is that we used for hydrogen production.
Ah, okay. Ah, you're collecting the hydrogen in gas.
Yes.
Oh, isn't it dangerous to have a lot of?
Oh, it's yes. It should be dangerous, but luckily and unluckily, technology for production of green biohydrogen from microalgae using photosynthesis is still in the developmental stage, and it doesn't reach productivities that allow it to explode. So we are lucky,
Okay.
and this little amount of algae won't produce enough hydrogen for us to burn our eyebrows.
Thinking about master students or people that are applying for their masters, would you have any suggestion or recommendation if they're choosing between UTU and another university?
I really like UTU. It's a nice environment. I haven't experienced any other university in Finland, but I experienced universities in Italy. UTU is great. It provides you with a lot. It provides me with a lot of information on how to move to Finland, how to live in Turku, opportunities for doing activities, social activities with other students. I wouldn't dislike doing my master here, and I'm really enjoying Turku. So if I have to do another master, I will consider UTU.
Okay, that's very cool. That's very cool. And as a last question, at UTU we're inspired by many things, mostly by science, but could you complete this sentence, get inspired by?
Cyanobacteria.
That's very cool. Thank you so much. Thank you so much for the lab tour, and see you in the next video.
Thank you, guys. It's been a pleasure.